In the second part of this lecture, we'll talk about the breakdown of fat molecule for energy production. So step 1 is lipolysis, which is the breakdown of fats into its respective building blocks, which are fatty acids and glycerol. And then in step 2, you have the activation of the fatty acid by the addition of a coenzyme A molecule. And once these molecules are activated, we can oxidize them in a process known as beta-oxidation to produce acyl coenzyme A as the end product of this reaction. And with acyl coenzyme A, they can enter through the Krebs cycle and eventually the electron transport chain for ATP production. Let's begin with step 1, which is lipolysis that happens right in the adipocyte or the fat cell. So in the breakdown of lipids, we will need the enzyme lipase, which is stimulated by the hormones glucagon, adrenaline, or better known as epinephrine. They are secreted when we have a low blood sugar level. So that happens during starvation, during stress of emergency, or during exercise. So with the enzyme lipase, it will break one triacylglycerol into one glycerol and three fatty acid chains. So these fatty acid chains will be released into other tissues, while the glycerol can only be metabolized in the liver. Remember, glycerol is an alcohol, and all alcohols are metabolized in the liver. So to utilize glycerol, we need to convert them into sugars, or the derivative of sugar molecules. And that is performed by a multi-step reactions. Step 1 is phosphorylation, where we add a phosphate group onto glycerol, forming glycerol 3-phosphate. While the second step is an oxidation step, where we convert the glycerol 3-phosphate into DHAP, or the dihydroxyacetone phosphate molecule. So I believe you could still remember this molecule. Where is it coming from? Well, it is coming from your glycolytic pathway, because DHAP is an isomer of G3P, which is on the direct pathway of glycolysis. So with G3P, it can either go into glycolytic pathway to produce pyruvate and then ADP, or it can be used in gluconeogenesis for glucose production. So we'll talk about glycerol, which is a product from lipolysis, but how about the fatty acids? Upon hydrolysis, fatty acids will enter the cytoplasm, but before they can be oxidized further, they will be activated by the addition of the coenzyme A. And to do that, we will need a large amount of energy, because that bond that's connecting coenzyme A with the fatty acid is known as a thioester bond, and it has a very high bond energy. So normally one phosphate group will be cleaved from the ATP, but for this case, two phosphate groups or the pyrophosphate groups will be cleaved from the ATP, forming the AMP. And not only this, the pyrophosphate group will then be further hydrolyzed into two phosphate groups by the water molecule, and that is catalyzed by the enzyme pyrophosphorylase. So this has made the reaction very favorable, as you will get more ATP hydrolysis to top up the production of AMP and more PPI. And the overall reaction is catalyzed by the enzyme acyl coenzyme A synthetase. So with that, you have got the production of acyl coenzyme A, or the activated fatty acid. However, there is a problem, because mitochondria are impermeable to acyl coenzyme A, but it can be transported into mitochondria when it is attached with a carnitine molecule. So what happens is we need carnitine acyltransferase number 1 to remove the coenzyme A from acyl coenzyme A and join it with carnitine, forming the acyl carnitine. So the first enzyme is encoded by the chromosome. Only then this molecule can be transported into the matrix by the protein translocase. And in the matrix, you have another carnitine acyltransferase number 2, this time encoded by the mitochondrial DNA to remove the carnitine, forming back the acyl coenzyme A. Then the acyl coenzyme A can proceed into beta oxidation while the carnitine can be recycled. So in the market, you will find this carnitine marketed as a weight loss supplement. That's because they claim that this can improve your fatty acid metabolism. But the fact is, for any healthy individuals, we are able to synthesize our own carnitine. And therefore, we don't really need to take this carnitine supplement at all. So in the matrix, the acyl coenzyme A is ready for beta oxidation, which happens in 4 steps. So I'm going to use the example of palmitic acid, which is a 16 carbon saturated fatty acid. And remember, the fatty acid is now attached with a coenzyme A because it is already activated. So there are 4 steps in beta oxidation. Step 1 is an oxidation step, which is performed by FAD. So it will take away 2 hydrogen atoms together with the high energy electrons, forming a double bond and one FADH2, which can then be used in the electron transport chain for ATP production. Step 2, you will have the hydration of this double bond by a water molecule. And with that, you have an introduction of a hydrogen atom and an OH group. While in step 3, you'll have the oxidation step to remove 2 hydrogen atoms from this carbon. So this oxidation step is performed by NAD, and upon the receival of the 2 hydrogen atoms, it will become NADH, which then again can be used in the electron transport chain for ATP production. So if you study about the nomenclature of fatty acids, the carbon that is closest to the carboxylic group is known as the alpha carbon, while the second one is known as the beta carbon. So you could see how the step 3 or the oxidation step happens right at the beta carbon, and for this reason, this step is known as beta oxidation. Okay, step 4 is cleavage. So in this step, we'll cleave out 2 carbon units from the fatty acid and at the same time incorporating a new coenzyme A to the remainder of the fatty acid. So as a result, you'll get the release of one acetyl coenzyme A and a shorter fatty acid that is again attached with a coenzyme A. So this last step is going to repeat many times until all fatty acids are converted into acetyl coenzyme A. So for the complete oxidation of a 16 carbon palmitic acid, you're going to get 8 acetyl coenzyme A, 7 FADH2, and 7 NADH. And all these compounds are high in energy because they can be further metabolized by a conventional metabolic pathway to produce up to 129 net ATP production. So just in case you might be wondering, what will happen to an odd number fatty acid, such as with 17 carbon, 19 carbon, and so on? We know that the beta oxidation of even numbered fatty acid produces multiples of acetyl coenzyme A, but the beta oxidation of an odd numbered fatty acid will produce one propionyl coenzyme A, which is 3 carbon, and multiples of again the 2 carbon acetyl coenzyme A. And that 3 carbon propionyl coenzyme A can later be converted into the succinyl coenzyme A, which is a common substrate in the Krebs cycle. Okay, here's an overview of the beta oxidation, which is a 4-step process. So in the first step, you have the activated acyl group, which is attached with the coenzyme A. So with the oxidation of the activated fatty acid, you have the production of a new double bond, which is then being hydrated by a water molecule to form or to add an OH group and a hydrogen atom. And then you have a second oxidation event by which you have got the production of a new double bond or a new carbonyl group, which is finally being cleaved by the process thiolysis to produce the 2 carbon acetyl coenzyme A and a shorter fatty acid that's again attached with a coenzyme A. So you are not expected to remember all these substrate names and enzymes, but given the name of the substrate and the reaction type, you should be able to predict the names of the enzymes. For example, the first substrate is fatty acid acyl coenzyme A, and this process is oxidation. So logically, the name is acyl coenzyme A dehydrogenase because dehydrogenation is an oxidation event, and so on. So to put you into perspective, a glucose molecule produces about 32 ATP from a 6-carbon molecule. So that is about 0.18 ATP per dalton, which is the molecular weight. But for a 16-carbon palmitic acid, you will get about 0.5 ATP per dalton of molecular weight. So that is more than double. And this explains why lipid is a much efficient molecule for energy storage.